Hello everybody, and first of all, thanks to ORS for having us to presenting here at the meeting. Yeah, MLab is focusing on glaucoma testing, on glaucoma diagnosis, and the clinical situation in glaucoma is quite clear. It's a very common eye disease leading to blindness if untreated, and even in the industrial world, we have 50% of severe glaucoma cases undetected. And as already Francesca pointed out, we need to find them earlier. And the problem is they don't feel the disease, they will not go to the eye doctor, so we have to find something else. And that's where MLab is coming into the game, because MLab is trying to develop a easy-to-use test device as a point-of-care device which you can use everywhere by everybody. It's, you don't need additional instruments, it's cost-effective, it's quick, and it could find those people who are still undetected to get earlier treatment. MLab was founded out of University of Mainz from my group in Germany. I'm heading an institute there for experimental and translational ophthalmology at the University of Mainz. And first, they develop a unique multiplex platform where they can use a little microarray on those test strips which are very similar to the ones you know from the COVID testing, but we don't test for one parameter. We can test up to 20 parameters in parallel, and makes it very quick, very easy, and with very low volume. So we can use this also for other things, and that we're always trying to do, but for glaucoma, this is also very useful, which I'll show you now. For the glaucoma testing, we are measuring autoantibody responses to retinal antigens, which are thought to play a significant role in the pathogenesis of glaucoma. It could be shown by us for the last decade in many, many papers in cell culture, in animal models, and also in clinical trials, that most of these antibodies, for example, against heat-shock proteins, will lead, for example, after immunization in an animal to a glaucomatous damage. So after a few weeks, they develop the glaucoma. And if you go to our human studies, which is a picture on the left side below, we can always find, and we did this in thousands of patients, a consistent antibody patterns with up and down relations of antibodies against retinal antigens in glaucoma patients. So this is shown in our work for decades in the scientific part. What we are doing now for the commercial test. For the commercial test, we bring this down to just 7 antigens on our lateral flow using testing for antibodies against some heat-shock proteins, for example, some actin crystallines. And these 7 antigen panel leads, this we can also test it already in more than 1,000 patients, to a sensitivity and specificity of more than 90%. Now we have a working prototype already of the test using the tear fluid, and this is what you see in the second graph, where you see the ROC curve just using, in this case, 5 antigens. We already get in tear fluid a sensitivity and specificity of more than 93%. If we go down, you see on the left side to 3 antigens, this will not be enough anymore. We lose sensitivity and specificity. So we need a minimum of 5 antigens. We use 7 to be sure and to get higher sensitivity and specificity. This is how the test is working. You have an applicator. With the applicator, you just take a little bit tear fluid from the eye. A few drops is enough. Then you put this applicator on the cassette. The tear fluid is running through, the microarray is developing. You see the dark points, which are the antibody reactions. They are read in, for example, by a smartphone reader application, detected, quantified, and then with a little AI in the app, a glaucoma score is calculated, telling the patients, you are suffering from glaucoma, go to the doctor for further treatment. The potential is huge because it's just the whole population older than 45 or 50 years with 1 diagnostic test per year, and this is also the business model. We foresee a yearly test for ongoing glaucoma disease in general population older than 45 years. If the glaucoma risk score is positive based on the test, you're referred to ophthalmologists to see the eye doctor. It will be provided through opticians, pharmacists, GPs, and later on also via OTC. Furthermore, in some countries, we got the feedback, for example, from India and China. This could be also directly interesting for ophthalmologists, because in those countries, many of the ophthalmologists don't have OTC or perimetry to do a real glaucoma diagnosis. So we already get the feedback from those countries that they are really interested. And you can see on the right side of the slide that we already have a strong IP protection in many countries. So what are the diagnostic modalities compared to other methods? Currently in the clinic, usually the glaucoma diagnosis is done by OCT and perimetry. For this, a big investment is needed. It is operator dependent to get good results. And again, it's not a point of care. IOP tonometry, as all of you know, is not good for glaucoma detection. It's good for monitoring disease, but kind of useless for glaucoma detection. So currently, we would be probably the only point of care device which can be used by everyone, everywhere, without any additional instrumentation. There's no further investment needed for the service provider. You will get quick results within a few minutes. It will be affordable. The test is probably less than 20 euro. You have a very high sensitivity and specificity. The marketing can be direct sales or through existing distribution networks. We have in our team already 2 sales experts building this up and preparing this. It also works for glaucoma subgroups. And again, for many countries in Asia, for example, or Africa or India, they are already interested to have this also for ophthalmologists. What is more in the pipeline, if this already is in the market, after the successful market launch, we will have a progression model of this test, which will be introduced then for ophthalmologists. This can be combined with imaging instruments like OCT, and it will determine the risks for the speed of the progression in the next year for this single patient. And what you can see here in the middle, for example, is a result of one of our clinical studies where we had an antibody measurement with our test many years ago, and then followed the clinical course of the disease of those patients. And you can see in dependency on the concentration of the antibodies, that as higher the concentration of the antibody was many years ago, the quicker the course of the progression was for the glaucoma disease. We are also discussing now to use additional microarray spots, which are still available. We can do up to 20 to 25 on those tests, to have a multi-disease panel, for example, not only testing for glaucoma and dry eye or glaucoma and AMD, and also going into the neurological diseases. We try now to use a microarray, for example, also in the tear fluid to test for Parkinson or Alzheimer or something like this, and this also can be matched by imaging instruments. So what is the status quo? We have large studies with more than 1,000 patients in SERA. We have a working prototype in tear fluid, and it's also backed up with a lot of clinical data. We expect the market launch in around 2 years. We're now in the next financial round. That's the reason for presenting here. What would be the action plan for the next 2 years? We have to get the certification for the company for production. We need to prepare the FDA or CE mark application, do the validation study, preparing the distribution. Our marketing expert is already preparing this, and having the launch not later than 2 years. So this is the MLab team. If you're interested in our company, please contact me and discuss. And thank you for listening.